Thanks for having me here. I'm actually very glad that my talk is right after LGC, the wonderful talks by Luke and also other specialists right here. So you will find my talk is actually very resonating with the previous talks and with all the information that you have received from the previous conversations. And I hope I show you a practical approach and our case basically showcase our company building an oligonucleotide drug discovery platform, and particularly from a chemistry perspective. First, let me introduce our company. We are founded in November 2020, and currently we have expanded our operations in Singapore, Boston, and Shanghai. And our mission is really to combine the proprietary chemistry and the novel biology to be able to accelerate the nucleic acid therapeutics development. And in today's talk, I will really show how we are going to develop the oligonucleotide drug. And from a traditional point of view, I think many people are very familiar with this workflow, starting with the target discovery validation, and then we will screen the small molecules, screen out a hit, and come to do the lead optimization. And once we finalize the lead, and then we move into the preclinical development. And for the hit screening process and the lead optimization, basically this is so-called pharmacophore development, right? So for small molecule, everyone is very familiar. We have to come up with a library of compounds and be able to really look into the modality that we are interested in and then be able to further optimize it. Then how are we going to do it with the oligonucleotide? It's a big molecule and it's a biopolymer. So it turns out actually you have to do very differently for this pharmacophore development. And this is how I concluded into, for the pharmacophore development in oligonucleotide drug discovery, it consists of 4 major components right here. The first part, based on the target that we have selected, you have to select the sequence that you find that matching the target and actually can be able to knock down the target. And then comes to will you be able to do the oligonucleotide synthesis that's generating the chemical matter? And then come to how are you going to modify it to achieve the optimum effect? And finally, do you have a good delivery system to be able to deliver your big molecule into the cell? So by looking at these 4 components right here, actually the 3 parts right there are the chemistry-driven platform development. And I'm going to focus on these 3 parts. And by combining all the information you have receiving from the LGC speakers. Of course, the core of the oligonucleotide synthesis is this very robust phosphoramidite chemistry has been there for over 30 years. And I think Luke has already given a very good introduction right there, so I wouldn't touch too much on it. Basically, it's a cycled reaction and it can be automated. And usually you have to detreat, basically it's 4 steps: detritylation, couple the monomers, and then do the capping to cap the filial sequence, and then further oxidize it. But for oligonucleotide therapeutics, like people have mentioned, we can use the phosphorothioate. So that means like you can replace the oxidation steps right there to sulfurization. And eventually, after a few cycles, based on the sequence you have decided, you will be able to complete the sequence by coupling all the respective bases together. And a general synthesis workflow would be starting from synthesis, and then you do the purification and do the analysis. The detailed steps, I think Luke also gave a very good introduction right there. I wouldn't touch on too much. But basically, you need a synthesizer, which we have, I think Luke mentioned about Mermaid 48, and we do have 48 and 182 in-house, and also other synthesizers from other brands. And for the purification step, you can choose to do a simple purification, like the evaporator, and also doing the simple C&D, and also do a quick desalting. But usually for our purpose, right, if you want to have some high purity oligos, you need to do the HPLC. So that's where the HPLC stage purify the oligos. And after generating the pure compounds, you need to do the quality check. So that's where the LC-MS comes in. Make sure that you have the correct compound with a good purity right there. So overall, it's pretty much a 3 steps workflow. And there are some issues right here. So I think Luke also touched on that. But I'm going to show particularly 4 impurities that we have seen related to oligonucleotide therapeutics development. So one thing, the first one would be insufficient deprotection. It could be coming from the insufficient deprotection of the base. So I think, for example, the IBU and the AC, they are difficult to deprotect. But this is very common for any oligonucleotide synthesis. One thing right here actually is related to universal support. So I think just now you have seen that people use the universal support to be able to generate lots of oligos without defining the first base. But actually what we have usually seen is that it turned out the universal support, the residue is not very easy to be deprotected. So you need to kind of have either longer time for the C and D, cleavage and the deprotection, or use harsher conditions. And the second part would be the depurination and the depurimidination. Depurination is very common for DNA synthesis if you are doing large scale or doing longer sequence. But for for the therapeutics, right, what we realize is quite common is the depurimidination. Of course, this is a hypothesis right here, which we found from a literature. But we found out that it's very frequent that we would get uracil, basically removal of a uracil. Basically, we will miss one U right there from the final compound. And the mass deduction you can see right there. So it's not concluded, but this is at least based on our best knowledge, the mechanism that we are having right now. And another common issue is the loss of diet. So we, as I mentioned, we do, usually people use PS, backbone, to modify the oligonucleotide sequence. However, it's actually very common for you to see that you are not getting the expected mass, but rather than -16. And that's actually caused by the loss of diet right here. There are a few reasons cause such issue. For example, insufficient sulfurization. It could also be a reagent quality issue. But if you see such, like -16, that will, at least you identify the problem is most probably from the loss of diet. Of course, there are other byproducts. The most common ones would be the failure sequence. If your coupling is not very successful, you will see missing one base or a couple bases. And also could be addition of the acrylonitrile. So this acrylonitrile is actually coming from the deprotection, right? It's a product from the deprotection. However, this acrylonitrile product can further react with the amino group and form this byproduct. But this one, it actually is very simple to remove. Basically, you just do a DEA wash on the CPG, and then this, I think the chances of getting this is actually very low. So one particular thing right here to solve the insufficient deprotection and depurination and all the other issues, one core thing is that we can't use just one simple method to be applied on any kind of sequence. Based on your sequence and based on the modifications you are choosing right there, actually you have to optimize the methods. It can be on your synthesis, like you change your coupling time. It can be your purification, you change your LC approach. And also a very important thing right here, maybe not many people know, you actually need to explore an optimum C and D conditions. So it cannot be too harsh that you are actually degrading your expected sequence. It can also not to be too mild so that you wouldn't be able to deprotect all these protecting groups of your sequence. So this is one important part right there. All right, so with all this knowledge, right, so in Cargene, actually, we have built a full range of oligonucleotide synthesis, starting from 15 nanomole to up to over 10 millimole scale. So we have our first puzzle piece right here. You will see at the end, we will have a full puzzle right there. And then the next part, if you remember from my first slide, I mentioned about the modification design's importance, right? Why is it so important? So I think you have seen this diagram before. So I will go through the RNA interferencing step to showcase this example right here, the importance of the modification. So for small interferencing RNA, the very important thing is you first need to be able to successfully let it go into your cell. And that means it has to be stable by traveling through all the bloodstream and coming to the cell and then coming to the endosome and finally be released as a small interferencing RNA. And the whole travel actually is very challenging for natural sRNA. So you have to modify it to make sure that it's stable all along the way. And then once it comes to escape from the endosome, and it will be further phosphorylated and be able to load it on the AGO2 to form this activated RISC assembly. And by doing so, your guide strand somehow has to be able to have to be phosphorylated and then be able to recognized by the AGO2. And then further on, with the activated RISC complex, it needs to be able to specifically recognize the target, the region on the mRNA, and enable the cleavage right there. So the whole process that requires the specific recognition on the targeted region, but at the same time to avoid any off-target effect. That means it's not recognizing another region on the mRNA, right? So this requires an optimum modification right there, so that it's not going too much, binding too much on the mRNA, but at the same time will be able to bind to the respective region and trigger the cleavage. So luckily, through over 20 years nucleic acid chemistry development, we do have a toolbox for modifications that we can use. So here I basically just list down quite a few different types of modifications. The first one will be the phosphonate modifications, which the most important one probably you have heard from all of us is the phosphoratase, but there are other kinds that you can utilize. And the second one will be the ribose modification. And all of these, you can try modify the monomer, be able to adjust its specificity of binding and the off-target effect right there. And another one is the base modification. To be honest, base modifications are less used. The key reason right here is simply the toxicity sometimes can be very difficult to control, but it doesn't mean it cannot be used. So people still explore that. So at the end, by having this toolbox, it doesn't mean if you apply it, you can just simply get what you want. The numbers of the modifications you use and the positions that you use them are also very critical to achieve the optimum effect. So in Cargene, we do have develop our own platform to be able to modify the oligonucleotides to achieve the respective effects. That includes our oligos can be stable. Secondly, our compounds are potent. And thirdly, that means it's very specific and it will be able to have less off-target effect. So we call this platform SMART platform, which is called basically short for selected modification patterns. And then coming to the next is actually a very challenging thing. It's the delivery system, which has been troubling the oligonucleotide drug industry for a very long time. So why is it very important? The key challenge, I think other speakers also mentioned a few challenges there. But actually the key challenge right here is really because the oligonucleotide, which has the highly charged phosphate backbone, and is also a very big molecule. It is very difficult to go through the lipid bipolar membrane. So people have been mainly utilizing two approaches. The first one, which we heard very frequently, is we used in mRNA vaccine, is a lipid nanoparticle. So basically, this is a carrier-based approach, what they do, they use the lipid to wrap up the capsulated the oligonucleotide inside, and then further be able to deliver it. So in this approach, right, it is effective. However, it's not very specific, and also the manufacturing is actually very challenging because it requires lots of formulation. So there are many people also exploring a more controlled way, which is the chemical conjugated way. So what they do, previously, people have been using lipids to conjugate it on oligonucleotide so that it can have generating a force when it comes to the membrane and be pulled in. Another way is more, so in this way is actually more passive, but then people found this more like a receptor binding approach. So by using this ligand receptor binding approach, it generates a pulling force, and the receptor can also go into the cell through endocytosis and then form an endosome. In that way, the oligonucleotides will be able to travel into the cell and then further release the sRNA. Among all these approaches, actually one particular one becomes very exceptional. So that's the GalNAc, trimeric GalNAc. In the next slide, I'm going to give you more information about the GalNAc approach. So it delivers very efficiently to liver, and it's very specific to liver targets. So if you look at the top left right there, so you can see the tri-GalNAc actually is conjugated on the sRNA, and it binds to the ASGPR, and once it forms this complex, it will be pulled into the cell through the endocytosis approach, and then further go into the endosome. And inside the endosome, because of the pH drops, it actually cleaves the linker which conjugates the GalNAc and sRNA, and eventually relieve the free sRNA inside the endosome. And through an unknown endosome escape approach, slowly the sRNA can actually be released to the cell and further trigger the following downstream silencing pathway. So through this way, then we will be able to successfully deliver sRNA to the liver cells and be able to trigger the RNA path. And there have been quite a few different kinds of GalNAcs have been developed so far by different drug companies. And you can see Alnylam actually came up with the first version, the tri-GalNAc, tri-antennary GalNAc. And then later on, Ionis also followed their approach to generate a tri-GalNAc. And actually, very recently, there are more people generating their own GalNAcs. For example, Sanofi is also having one, and LGC has also showcased theirs. And us, we also developed our own proprietary GalNAc right here. And our GalNAc delivery system has already shown very good in vivo knockdown efficacy and duration. And the safety profile also have been supported in vitro and in vivo. So eventually, you have seen our three puzzle pieces. What happens, we do have the full range of the oligonucleotide synthesis and the smart pharmacophore modification design and the proprietary ligand development. And with this platform, we are actually confident that we can combine our proprietary chemistry platform together with our novel biology platform to continuously generating more exciting oligonucleotide drugs right here. And a brief intro right here about our biology platform is that we do have the target discovery and validation, and we have an OSCA bioinformatics-driven sequence selection platform, and we also have a rational assay cascade to be able to screen out and also identify the leads. So this is our whole platform, and we aim to accelerate oligonucleotide drug development, not only in Singapore, but also globally. So thank you very much.